After you have contraction, you have to have relaxation. So I want to walk through what the steps are to have relaxation. So this is going to occur after everything that previous video happened. What we need to do is get rid of the signal, initial signal, and also get rid of calcium. So this here is our neuromuscular junction. Here's our myofilaments. And what could this thing be? This is our acetylcholine receptor. With acetylcholine, you already know that. What could this thing be? Well, you know how neural signals are terminated. There's 3 different ways. For the neuromuscular junction, the enzymatic degradation is really important. There's an active enzyme. This is the only thing that's going to cause relaxation. We're not going to wait for ACH to diffuse out of the neuromuscular junction. We want to break down ACH so we can control when relaxation happens. So this is called acetylcholine esterase. ACHE is the abbreviation. Acetylcholine esterase is an enzyme that breaks down acetylcholine. It's going to terminate the signal in the neuromuscular junction itself. So that's one thing that needs to happen. When that happens, we're going to have a decreased signal of calcium release. Instead of calcium flowing out of the sarcoplasmic reticulum, it's going to go back into the SER. How does it do that? Levels are already high in the SER. We need to pump it back in. We are going to use label this. Here's the SER, is this blue stuff along here. We're going to use a calcium ATP pump. Calcium ATP pump. It's going to use ATP to move calcium back into the SER. It requires ATP because we're pumping calcium against this concentration gradient. Calcium is still high in the SER. When calcium is pumped back into the SER, it's no longer bound to troponin. Here is our troponin. Calcium is no longer on there. That causes tropomyosin to move back and cover those myosin binding sites on actin. So actin and myosin can no longer be bound when calcium isn't present. A loss of calcium ions from troponin returns tropomyosin to its starting position, and that muscle is going to go back. The sarcomeres are going to lengthen again and no longer be overlapped. They'll be not unoverlapped. Our A band and I band are going to be back to their resting positions. Relaxation. This happens very quickly. So what we've just done is describe the sequence of events involved in skeletal muscle relaxation. And these are the terms that I used for that.